for being here. Today we have a speaker, Cristina Romero Castilla. She did a degree in chemistry, and then she did a PhD here at the ICM. Then she was for 5 years as a postdoc in different places, mostly in Miami and then in Vienna, in Austria. Her work is mostly about the carbon cycle in the oceans, and she has been working lately in the plastic, especially on the role of the microplastics in marine ecosystems and how the bacteria can participate in this problem, perhaps in degrading these substances. Cristina, she has got several awards, Women in Science, an International Rising Talents in 2020 from the L'Oréal UNESCO, and also the and she also recently published a book, Anthropocene, Anthropoceno, which I think that it's a very, very interesting book. And we are very glad to have her here for this talk. So, Cristina.
Thank you. Well, I don't have a mic, just this, no? Okay, okay. Okay, thank you all for coming. Sorry, I have to start with a very ugly picture because it's nice to work in marine science because we always can use very nice pictures in our presentations. But since I'm going to talk about garbage and plastic, so I have to start like this, and it's not very pretty, but it's what it is. So I'm going to talk about the plastic degradation, in the impact that plastic degradation has in the biogeochemistry of the ocean. So in 2020, 367 million tons of plastic were produced globally, and out of that amount, less than 4% reach the ocean every year. So less than 4% can seem not very high amount, but when we are talking about 300 million tons of plastic, it's a lot. So more or less up to 13 million tons of plastic reach the ocean every day. And here are the most common plastic that we use in our daily life. Most of them are the most found in the ocean as well. We have LDPE, low density polyethylene, that is used usually to make plastic bags, for example. Then we have high density polyethylene that is used to make a bottle of detergents or shampoo, things like that. Then we have polyethylene that has been used, for example, to make CD cases. And we have also expanded polyethylene that here we call polyespan, that is used in food trays, for example. And polypropylene that is used also for food containers, among others use. Now, recently, more and more, it has been an increasing interest to find new materials to substitute this plastic made from petro-based plastic. And now there are many, there are some options of biodegradable plastics that has been recently increasingly used. One of them is the polylactic acid, or PLA, that is biodegradable and compostable. But this plastic can only be compostable, can be degraded only at temperature higher than 50 degrees and at a special condition of humidity that only are given in compost industrial plants. So they are not biodegraded in the ocean or in the field. This plastic, I have seen it so very more increasingly now because it is being used as substitute of single-use plastic items. For example, now here in the festivals in Barcelona or in Feste Majors, or many times they give you this plastic instead of the conventional plastic. The people just discard it as the other one without maybe think that it also has an impact in the environment. When the plastic reaches the environment, it starts to degrade, and there are several factors contributing to the degradation. The most important is the sunlight degradation, but it also can be erosion, oxygen, high temperature. All these make the plastic degrade. And when we talk about degradation, it doesn't mean that the plastic disappears. It means that it starts to present scratches, brittlement, and it can fragment, break into fragments. That when those fragments are smaller than 5 millimeters, they are called microplastics. But they don't disappear. We all know about many problems of the plastic, about animal entanglement, animal ingestion, that plastic is reaching us through the food, through the water, and how bad the plastic it is. But there is something that is not that known, and it's about the chemicals that plastic can release into the media. For example, plastic, the polymers are not pure. Usually the plastic we use, it has additives. It has like antioxidants, plasticizers, colorants, to give the plastic the properties required for its use. And these compounds are not bound to the plastic with strong forces, strong bonds. They are linked bonds with weak forces like van der Waals forces. So they are very easy to unbound and be released into the media, into the aquatic media. So when the plastic reaches the ocean, for example, it can lose their additives in the process called leaching, but not only the additives, also it can experience depolymerization. This is the process by which the polymer is broken into monomers or oligomers, and those are also released into the media. Other property of the plastic is that it can also absorb the organic matter that is in the media. For example, persistent organic pollutants can be taken by the plastic and then absorbed and then released into the media later on when the conditions change. So here we have the carbon cycle that I'm going to explain very briefly. And overall, I'm going to focus on this pool of the carbon cycle. That is the dissolved organic carbon. This is a pool of carbon that is making part of molecules, organic molecules. And all this pool of molecules make the dissolved organic matter pool, DOM. But we don't have a way to measure that DOM because the composition is very diverse. And it's not easy to measure it. So actually, we just know like a 10% of the chemical composition of this pool of organic matter. But to measure how much of this matter we have, we measure the dissolved organic carbon. So this is that's why it's also called DOC, or dissolved organic carbon. So this carbon is mainly produced by phytoplankton in the euphotic layer, and is consumed. The main thing are marine bacteria, which consume this carbon, so it's at the base of the trophic web. But there is a small fraction of this carbon produced by phytoplankton that escapes the microbial loop, and it has been accumulating in the ocean for centuries, for thousands of years, until we got an amount of 662 gigatons of carbon. And this amount is very similar to, or the same magnitude to the carbon that is making part of the carbon dioxide in the atmosphere. And since both pools are linked, any single change in one of them will affect the other and will affect climate. And I'm very interested on this pool of carbon, and it's the topic that I have been studying since my PhD, because there are many unknowns about this pool of carbon, and it's very important for the carbon cycle. So now I study the sources, the sinks, how it's distributed, and all of that. So one of the things that I found studying this was that the plastic could be a source to this VOC carbon, to the dissolved organic carbon. So plastic could release this carbon. So in a previous study, we did a series of experiments where we found that this happened like that. No? If we do a zoom to this, so we saw that how plastic is releasing these chemicals that we could measure with the dissolved organic carbon measurement. And this leaching is enhanced with the sunlight radiation. And in that study, we did an estimation, knowing that 13 million tons of plastic reach the ocean every year. We estimate that 23,600 tons of DOC can be released from plastic every year. We also found that marine bacteria were able to degrade those chemicals. Actually, the 60% of the dissolved organic carbon released by plastic could be consumed by bacteria. Knowing all this, and after this study, other studies have been studying the leachates of how the plastic leachates affect the microbes in the ocean, or also in other aquatic systems. They have found, for example, that they can also affect phytoplankton, And other studies have confirmed our results that that all these chemicals are stimulating marine growth, marine bacterial growth. But there are many open questions about this, about how these leachates affect the carbon cycle, the biogeochemistry of the ocean, and the microbes. And also I had the question to know which were those bacteria that were degrading those chemicals, okay? So these are all these questions is that we're going to answer here that through a series of studies that we have done and have been published this year. So to answer all that, we did experiments in two phases. So there is a first phase that is abiotic, in which everything is sterilized and there is no bacteria here, in which we place some plastic pieces in seawater that is sterilized in quartz tubes, and then we irradiate it with UV and visible radiation in a solar stimulator. We irradiate it usually for 6 days. And then we study the chemicals, so the dissolved organic carbon of the leachates, okay, and the characteristic of that dissolved organic matter that is released. And we also do dark treatment of plastic, the same but in the dark, and also controls without plastic. We always do control of the light and also the dark, and everything is done by triplicate. And after having this and studying the leachate, the chemical composition of the leachate, then we do a second part of the experiment in which once we have the leachate, the plastic are removed, and then we add a natural bacterial inoculum that we filter through 0.8 to remove traces, and we incubate this in the dark during a few days until bacteria reach the exponential phase. And once they reach it, then we stop the experiment and we measure again the same chemicals, the dissolved organic carbon, the dissolved organic matter that we have left, and we also do some analysis, microbial analysis that I explain you later. We also follow this experiment analyzing microbial abundance every day to follow the curve and know when they start to grow, they stop to grow. So other thing that we do to measure to know more about the dissolved organic matter is released by plastic, is that to measure its fluorescence, because we know that I said before that it's very difficult to study the chemical composition of the natural dissolved organic matter, but we have a way like a proxy to study that composition, and it's through its fluorescent properties. So we know that the natural DOM has a fraction of it has the capacity to absorb UV and visible radiation, and that fraction of the DOM is called colored dissolved organic matter. And a fraction of it is able to fluoresce, emit in form of fluorescence, the absorbed radiation. And this property of fluorescence is that we use to know more about the chemical composition of the DOM. So in the same way that we use it for the natural DOM in the water, in the ocean, we also use it to know more about this from the plastic, the material released by plastic. So to know that, we run a series of spectra, fluorescent spectra, at different excitation wavelengths, but we recover them at the same emission range. And we build, putting one next to each other, we build this 3D matrix where we have excitation, emission, and the fluorescence here. And if we look at this from the top, we have something like this, no? The excitation, emission, and here the fluorescent intensity. This is just to show you how it looks like the natural DOM from the ocean. We have an area where the protein likes substance used to fluoresce. And this fluorescence is due mostly to the tryptophan, but also can be due to the tyrosine that is fluorescing a bit shorter wavelengths. And then we have this area where the humic-like substance fluoresces. And usually we find these three peaks. And depending on the position of these peaks, we know the compounds are more or less aromatic, for example. We can also know the origin of the DOM, knowing the fluorescence as well. So, and this is the structure, for example, of typical amino acid, humic acid, that would be giving fluorescence in this area, just to show you the structure. So these are the plastic that I introduced you before that we use in all these experiments. And the first question we have was regarding to the degradation stage of the sample, of the plastic. Because the most of the study that has been made with leachates has been made with new plastic that is not degraded. But the most of the plastic that is found in the ocean is already degraded, it's weathered. It has been experienced to sunlight, it has present scratches and everything. And we wanted to know if that aged plastic that is more real that we found out in the ocean would be releasing more or less dissolved organic matter than the new plastic, and if the material release was more or less bioavailable to microbial uptake. So we used two types of plastic, LDPE. We used pellets for these pellets that were new, and then we used aged plastic that we collect on the beach. The composition of this plastic was 65% of polyethylene, 30% of polypropylene, and 5% of PVC. So with this, we generate the leachates in the way I showed you before. Well, here is just to show you in the electron microscope how the surface of each one looks like. This is the new plastic, and the aged plastic presents a lot of scratches, so it's very different. So here are the results of the material released by these two types of plastics. So here we have the VOC leached by the plastic. This is the irradiated treatments and the dark treatments in the gray. And we have the LDPE here and the aged plastic. And you can see that the aged plastic releases much more VOC than the new plastic. And if we zoom this part, we have that the irradiated sample release much more VOC than the non-irradiated. And we did those estimations that I showed you before about how much VOC is released into the ocean from the plastic reaching the ocean every year. But we did those estimations using the values from experiment with new plastic. But now that we have the values of the aged plastic, that is the one that is found in the ocean, we found that this amount would be 57,000 tons of VOC releases per year for the plastic entering the ocean, plus the VOC that is released by the plastic that is already there, because the plastic is not stopping to leach. So it's more than this, probably. And here we have the result of the leachate of both, this is only the aged plastic, because in the new plastic, or the LDPE, we didn't find any clear peaks of fluorescence. So here are the fluorescent matrices. I have left here the natural F-dome fluorescent matrix just to compare, but these are the ones from the experiments. These are from the leachate generated in irradiated condition and those generated in non-irradiated. And in both, you can see that the plastic release substance fluorescing in this area, which is the area where the protein-like substance fluoresces in the natural DOM. Of course, these substances are not, from the plastic, are not proteins, but there are other substances fluorescing in the same area than proteins. That could be, for example, polyaromatic hydrocarbons or phenols or others. Probably these are polyaromatic hydrocarbons. And notice also the difference of the scale. So here the intensity is higher, the fluorescent intensity. And this could be because the one that is exposed to radiation has photodegrade the fluorescent substance here. So probably it could be the same amount of fluorescent matter, but this has been photodegraded, so that's why we have less signal here. And then we saw if those fluorescent material released by plastic were bioavailable by microbial uptake in the second part of the experiment. And we have here, again, the irradiated leachate and the non-irradiated. And the negative values are fluorescent that has disappeared, like compounds that has disappeared. So we can see here that in both, bacteria has consumed this substance that fluorescent in that area. So this fluorescent DOM was bioavailable to microbial uptake. Then what about other plastic types? And what about biodegradable plastic? So to answer this, we use also these plastic types, including a biodegradable plastic, because we want to compare the biodegradable with the other, more petro-basic plastic. So here we have the citation emission matrix of the expanded polyestyrene. This also shows a fluorescent signal here, also in the area of the proteins. In this case, the irradiated, this is the irradiated leachates, were not lower in intensity than the dark ones. So probably the material released by the EPS was not that easily photodegraded. Regarding to the other plastic, we found a fluorescent signal, but we didn't find any peak like this, no, a clear peak like this. So that's why I'm not showing these matrices. So probably because the new plastic is releasing less amount of this material and also fluorescent. However, the EPS is a plastic that is faster, leaching, and probably that way is present this fluorescent. So here also we have the amount of DOC and fluorescent DOM from the leachate of these 4 plastic types that I showed you before. So this is the amount of DOC leachate and the light and dark. We have here that the LDPE, the low density polyethylene, was the one releasing more amount of DOC, followed by the EPS. Okay, that was here. And then in all of them, the irradiated was higher than the dark one. And interestingly, the biodegradable plastic released less amount of DOC than the rest of the other plastic. So we already know that this plastic was not biodegradable in the condition of the ocean, but I would expect maybe to have more leaching than the others, but it wasn't. And regarding the fluorescent, the one presenting higher fluorescent DOM was the EPS, and the overall in the light in this case, and the other were present like a small fluorescent, and the PLA, the biodegradable, showed negative values, probably because it absorbed the fluorescent material that could be present in the seawater. And regarding the viability of these leachates, of these plastic types, we found that the highest consumption of DOC took place in the EPS treatment, in the irradiated one, and the other present similar results, including also the biodegradable PLA. So I would have expected also that the chemicals released, the organic matter released by the PLA would be more bioavailable to microbial uptake because it was a biodegradable plastic, but it wasn't. Regarding the protein-like, the fluorescent, in the protein-like region, we found that the EPS, the fluorescent compounds released by the EPS were consumed by bacteria, but the others, it wasn't. So it seems bacteria produce a protein-like substance because we know that they also can produce it, and the production was higher than the consumption probably, and that's why we see here a production, net production. Then how plastic leachate affect microbial activity. So here we call about microbial activity to the capacity of the bacteria to synthesize proteins. To answer that, well, I collaborate with Marta Sebastian, who is here somewhere. So yeah, she's the expert on this technique that is called BONCAT, the orthogonal non-canonical amino acid tagging, that consists in adding an artificial amino acid to the bacteria. So once they take the amino acid and synthesize the proteins, we add a dye that is bound to the amino acid, and it's going to have fluorescence. And through epifluorescence microscopy, we can see those cells that are fluorescing, and depending on the intensity of that fluorescence, we can know if they are more active or less, actively synthesizing proteins. This technique that uses click chemistry, that if you remember, this click chemistry has been awarded this year with the Nobel Prize. So well, if you have further questions or the details, so the super expert is Marta here. So to answer this question, we use 2 types of plastic, a new one with LDPE in form of pellets, and an aged mix of plastic as well. Okay. So here are the result of the activity. These are the bacteria that were grown in the leachates, okay? Leachates of low DPE, new one, and aged mix of plastic. and the control without plastic. So we have here the number of active cells in each treatment, and here the percentage of active cells in each one. So here we have the inoculum, then this is the result after 1 day of incubation, and this after 4 days of incubation. And we have those activity of bacteria grown in irradiated leachates and in non-irradiated leachates. And we found that in the treatment with the aged plastic, the bacteria were more active after 4 days of incubation, both in light and dark. We didn't see significant differences here. And then in the percentage of active cells instead, the highest percentage took place after 1 day of incubation. But here there are less differences regarding the control between the plastic and the controls. Just in the day 4, in the dark, the aged plastic also presents a significantly higher percentage of active cells after 4 days. Then we wanted to know also the structure and activity of marine bacterial communities responding to plastic leachate. And for this also I collaborate with Marta, again with the bone-cut technique that was combined with cardfish. It's another technique that allows the fluorescent identification of phylogenetic groups of interest. And to do that, we have to use probes which are specific for each phylogenetic group. It's also an hybridation taking place with fluorescent at the end, so you can know if there are bacteria of that phylogenetic group that you are using, that you are targeting. So for this, to answer this, we use this different type of plastic, but all of them were new. Okay, and you have seen that there are differences between new and age already. Here is just to show how it looks like this methodology. When we stain bacteria with DAPI, there is a stain that stain the nucleic acid, so you can see all the bacteria here fluorescing in the microscope. Then when you use the bone cut, you have the same bacteria, but just only those that are active, synthesizing proteins. And then with the cardfish, you have the same picture, but only showing those bacteria that are, in this case, Alteromonas. That is the phylogenetic group that we tried here. No? So for example, here you can see these circles. These are bacteria that are here, but they don't appear here. So these are bacteria which are active, but they were not Alteromonas. They were other things. So we tried different probes of different phylogenetic groups to see the composition. So here these are the results. So we have here two different experiments that we couldn't do at the same time because the logistics. And so we always use LDPE like a standard that was used in both. And then we use polyestyrene in this case, and here PLA, the biodegradable. We have the irradiated, the non-irradiated, and here the number of active cells. Here, so this is the number of active cells, and this is the contribution of the phylogenetic groups to this activity. Okay, so we saw that here that the LDP was the treatment where more active cells were. Here there are some, both plastic, PLA and LDP were more active than the control without plastic. And in some cases, there were no differences between the control and the plastic. And then we have that the predominant phylogenetic groups were Alteromonas, Rossobacter, and other gamma protobacteria. Here were more abundance of the other, and here more the Alteromonas. Okay. So in this experiment, that happened also is that the release of DOC by this new plastic, was low. So probably that's why we didn't see many clear differences or the differences that we expected in this. Or also that maybe the differences are at a more taxonomical level. So that's why we did also this. We also wanted to know the taxa responding to different leachates, and for that we did the 16S amplicon sequencing. This also in collaboration with Marta. Marta did all the microbiological part here. So, and this is, we use LDPE, new LDPE, and H plastic. These results are not published yet, but I show you just to show a little bit that there are some differences. Here, even if it seems that the community composition is pretty homogeneous, when we look at the abundance of each taxa here, we can see some differences. So here, this is a heat map, and here we have the yellow are the irradiated treatment, the dark are the non-irradiated, and the different colors of green are the different treatment, the different plastics. So here is the aged plastic, here is the LDPE, and the lighter green is the control. So the samples are clustered based on the community composition of the sample. And then also here, the different colors are the different taxonomy of the taxa. So here, that we found is that, for example, here we have taxa abundant in the original inoculum that do not thrive in the treatment. But then here we have taxa abundant in the original inoculum that thrive in control and non-irradiated LDP. More interestingly, here we have taxa that respond only to aged plastic that was the leachate that were previously irradiated, or here taxa that respond to the aged plastic, both previously irradiated or not. So there are some differences on this, and we are now working with these results. And just to end, we have a last question that was, is the DOM released by plastic altering the pH of seawater? And to answer this question, we collaborate with Carlos Perejero and Eva Calvo to know about this. And we try different types of plastics, including new plastics and also aged plastic. And this is come from because we already know that the sunlight radiation, the photodegradation of the plastic was releasing carbon dioxide and also dissolved organic carbon. So we were wondering if that carbon dioxide could be, or something released in the dissolved organic carbon pool could be affecting the pH of the water. So we did several experiments, only the abiotic part that I showed you at the beginning, this kind of experiment, just to see if the pH was decreasing after the irradiation of the sample during the generation of the leachates. So the stippled bars are the time zero of the experiment, and the last bar in every experiment is the control without plastic. And then we have also some new plastic like this LDP here, and this EPS and PLA biodegradable here. But we also have a different type of aged mixed plastic here. And you can see that in all the treatment with the aged plastic, the pH decreased in all of them. While in the new plastic, the decrease was very light or sometimes not significant. Just in the EPS, the decrease was significant but very, very slight. And then in the dark, we see that there are no significant differences between the amount treatment of new plastic, EPS and PLA, with the control and the time zero. And in this experiment, we just saw a difference with the aged plastic, that even in the dark, decreased the pH a little bit. Putting all these results together in a plot showing the pH at the end of the experiment versus the amount of VOC leached by plastic, we found that there is a correlation. That is exponential correlation, exponential equation, showing that the pH decrease was depending on the amount of VOC leached by plastic. So we found that this curve reached a plateau at 7.5 units of pH, and that the highest decrease experienced was of 0.5 units. And this decrease this change in the pH is comparable to the projected for the end of the century in the surface ocean due to anthropogenic emissions. So in the worst scenario, it has been predicted that the pH will decrease 0.5 units. Of course, these results, we use a concentration of plastic that is found in the ocean in very polluted areas. But it's true that if the water is moving and is with the waves and the movement and the mixing, so this effect can dissipate. But in very polluted area with water in calm, this acidification could be important in the surface microlayer or in the first layer of the ocean, and could be affecting the microbes here. Then we wanted to know also if this decrease in the pH was due to the production of CO2, or it was also due to the production of organic acid released by the plastic. And to do that, we measure the alkalinity of in one of the experiments. So, and we also estimate the CO2 through the alkalinity and the pH. We estimate this CO2 as well, how much CO2 was produced or the variation. And we found, so we knew that if the decrease of pH is due to the production of CO2, the alkalinity shouldn't change. But we saw a decrease of the alkalinity in the aged plastic treatment. That means that the plastic is not only producing CO2, that is also seen here, but it's also releasing organic acids that are contributing to the decrease of pH. So to conclude all this, the result of all these studies, we know that aged or weathered plastic release an amount of VOC that is 2 orders of magnitude higher than new virgin plastic. That plastic release DOM that can be traced through fluorescence, and this is something that we want to explore now. I'm collaborating with Marco Talone because we want to see in situ if we can track the plastic through the fluorescence of the organic matter that is released. Then we also saw that the fluorescent material released by plastic is bioavailable to microbial uptake, and that the DOM released by biodegradable plastic is not more bioavailable to microbial uptake regarding to that from petro-based plastic. We also found that plastic leachate enhanced bacterial activity, especially after the sunlight exposure of plastic. And plastic photodegradation contributes to seawater acidification. And to end, that plastic leachate alter the biogeochemistry of the ocean, at least of the surface in very polluted areas, as well as the microbial community composition. And I want to thank all the co-authors of these studies. Some of them are in one paper, others are in several, but it was Rebecca Mayengo who did her PhD with me and now is working as lab technician. Stephanie Brinstill, who also did her master's student. Marina Briones Rizzo and Marola Janet, who were her intro students. Arturo Lucas, who was lab technician also. Marta Sebastián, Pepe Álvarez Salgado, Eva Calvo, and Carlos Pejero, who you already know, who are researchers. And also the funding to the Plan Nacional project for young research, the Confuturo also, and ISDIN. And then I wanted to end with a nice picture, not with the garbage picture, just to thank you for your attention.
Thank you, Cristina, for this nice talk. We have time for questions.
Thank you, Cristina. It was really interesting. I have many questions because I really enjoyed it, but I'll try to keep it short. I wanted to ask first, how is the plastic distributed in the ocean regarding depth and geographical gradients? Is it all concentrated in the surface, or are the particles in a size range enough that allow them to sink to the deep ocean, or is it everything?
The thing is that the most of the sampling of plastic are made in the surface because it's the floating plastic. So we don't have that much information about the water column. I have seen maybe just a couple of papers about the water column, and some of, they were not very, I don't know, because they have maybe just one sample, not many profiles. So I don't know if they are very reliable to, of this. There is that in the bottom there is also a lot of plastic because it's eventually end in the bottom. But the distribution in the water column is not that known.
But it does sink in principle?
Yeah, it can sink depending on the density of plastic. Yeah. some plastic, like the PET, for example, or some of them, or the PVC, they sink because they are more dense than water. The polyethylene and polypropylene, for example, they float. But even those that float, if they are colonized, at the end they can also sink as well. Because they can be colonized by microorganisms and they can sink as well.
And in these experiments where you collect the plastic from the beach and you inoculate it, the water medium, has conditions that are typical of the surface, like few nutrients, but
we use surface water from Blanes, but we also add nutrients to be sure that the bacteria were not limited by that.
And the plastic that you're putting in there that you collected in the beach, the composition of it reflects what you see in the ocean at a global scale, or is it very specific to what you find at beaches?
The composition of the aged plastic was mainly polyethylene and polypropylene, and these are the two plastics more found in the ocean.
Nice. And finally, in the cultures, do you see the bacteria in kind of like a pelagic lifestyle, or they are attached to the particles or growing on it on biofilms or something like that?
No, because the thing is that we never put together the plastic pieces with the bacteria. So we use the leachate. So we remove the plastic pieces after having the leachate, and then we put the bacteria. The most of the people study bacteria attached to the plastic, but here is to study the degradation of the leachate.
All right, and finally, if the concentration of microplastics in the ocean high enough for it to significantly contribute to the process of ocean acidification, or is it just because in the experiment the concentration of the plastic is so high?
So the concentration, we also compare, the concentration is similar to the concentration of some polluted areas, very polluted areas. So it was that concentration, and even higher is found in nature. But it's true that I said, since the water is moving, so that effect can be dissipated. So so depending on if the water is calm and it's very polluted, can have an effect on the microbes, for example.
Awesome, thank you very much.
Thank you, Cristina. It was a really interesting talk. I also have lots of questions, but I will only ask you two. And they are related to the last part, the contribution to the decrease in pH. So I was wondering, how did you do the treatments? If it was a closed container, therefore the CO2 couldn't escape, and that's why maybe it is contributing even more to the decrease in pH. And the second one is that if you characterize the buffering capacity of the water that you are using, because maybe you are using, if you say Blanes, I assume it's a highly buffered seawater already, high alkalinity. So if you have an idea what will happen if it's seawater that has less buffering capacity, I don't know if there is a correlation between the high polluted areas and the buffering of those waters.
Well, regarding the first question, we did it in closed flask. So and we measure through the colorimetric technique, we measure the pH, but that was measured by Carles Pelejero and Eva Calvo group. I think I have seen Carles here, no, or no? Because I want him to answer you the one about the buffering, because this I don't know. This about the buffering of the water, so it's a pity. I wanted to know too.
Thanks, Cristina. I wanted to ask you, when you collect the plastics from the beach and use them for the experiment, how do you deal with the bacteria attached to them? Do you autoclave it? Or
no, we clean them, but before that we checked them at the electron microscope, and I was so surprised that there were no bacteria. So probably because at the end, I don't know, bacteria don't like they were in the beach, in the sun, since bacteria don't like that. But I would expect to have a biofilm or something, and there was nothing. But still, we did a process of cleaning using hydroxide peroxide to remove all the organic matter. So we cleaned it, and we were sure that there were no bacteria.
And this does not change the structure of the...
No, we follow a protocol that it has been used in other studies. It was very diluted concentration of the peroxide, so supposedly, no, it doesn't alter.
Okay, thanks.
Well, thank you, very nice. I have two questions. One is related to the pH experiments, that if you check at the beginning, at the end, that there is not bacteria there, they were...
Yeah, they were sterilized because we did it, this was the first part of the experiment that we do to generate the leachates, and it's always abiotic. And we always measure the bacteria at the end of the experiment to be sure that they didn't grow.
And the other is, you say that you were adding nutrients to be sure that bacteria are not limited. But this is what they are doing when there is some oil spill, no? They put nutrients, because with nutrients you can degrade more the petroleum, and I guess it will be the same case for plastics. But then this is not the normal conditions in the surface, which are...
Yeah.
So maybe the surface without nutrients, bacteria are not...
Yeah. So I think it would be nice to have another experiment without adding nutrients to answer that, because since we wanted to answer the question of if bacteria are able to biodegrade this, so we add the nutrients to be sure that they didn't stop to biodegrade it because the nutrient limitation.
And then you cannot extrapolate to nature.
Okay, because it's I see, because I see. Yeah, because if they are in an environment that they don't have nutrients, no, they could not degrade that anyway. Yeah, that's true. Yeah, I think it would be nice to do an experiment also to check that.
Yeah, usually experiments, you know.
Yeah, they cannot,
Thank you for such a nice talk, Cristina. If I'm not wrong, you showed that the expanded polystyrene released like protein, like organic matter,
fluorescence,
right?
yeah.
Fluorescence. Could it be the styrene itself, or oligopolymeric styrene?
Yeah.
Because I checked quickly while as you were talking, and the fluorescence of styrene kind of resembles that of this tryptophan peak.
It's not only additives that are released, but also the oligomers or the polymerization can happen, and also we can see also the styrene, exactly. Yeah, it could be.
Okay. Yeah, and if you remember, we saw the styrene being released.
Exactly. So, because just to know, we are doing some experiment with Rafael and Charel to measure the volatile organic compounds of the released by plastic also, and we have found this very interesting.
Hello, Cristina, thank you. And I had a curiosity. Well, I understood that the DOM, the organic matter release, should depend on the amount of additives that the producer adds, basically, right? And depending on different plastics, they may add more or less additives, right? So that's different, even the same plastic may have more or less additives. And so I'm wondering whether you could measure the amount of additives before they becomes dissolved organic matter. So inside the plastic, can you measure that?
We haven't measured, but it's something we want to know with Berta, who is there. She's going to start next week with me, a postdoc, Margarita Salas. And she has done her PhD with additives of the plastic, analyzing them. So one thing we wanted to know now is which additives are released now, how they are.
Even before they're released inside the plastics, that's what I'm wondering.
I have read some studies that they dissolve the plastic and analyze the additives. That can be done too. Before and then you can know what to look for.
And also, just a curiosity, in the emission absorption spectra, the one that you showed, how can you discard the presence of amino acid in that peak? Like, is there any procedure
because we know that the plastic has no amino acid composition, so cannot be the plastic is releasing amino acid.
But if you treat them with bacteria, then...
no, but that was before adding the bacteria. That was during the phase that was abiotic. So no.
Then you measure it again, maybe after adding the bacteria.
I know what you say. Yeah, we saw. Yeah, then there we saw a consumption. I don't know if you remember in the aged plastic, a consumption of that peak. But for the other plastic, some of them we saw a production of the fluorescent in that protein-like region. And we thought that it was because bacteria were releasing more than they were consuming. Because usually bacteria take that protein-like substance because supposedly are labile. But they can also produce it. So in this case, I think that they produce it.
And before adding the bacteria, do you exclude the possibility of contaminants, like protein contaminants that I don't know how easy it can be that.
Yeah, but we measured the fluorescent at least and the VOC before and after, and we made here was the subtraction before and after, so we can exclude all that was in the water just to see the variation.
I also have a question. I think one of the results that you had is that the aged plastic produces more leachate than the new plastic, which for me it's a little bit counterintuitive. So what would be the cycle of this leaching of the plastic?
Yeah.
Will it be infinite? Or
No.
how it will stop to leach the very old plastic or something like that?
So, well, nobody knows because nobody has done that long-term experiment, like to know when they stop to leaching. But yeah, it makes sense because the plastic usually has some antioxidants and compounds, additives, to avoid the degradation. And those are lost with the degradation, with the weathering, with the radiation. They start to be lost and they stop to protect the polymer. So it can release more. So there are other studies that they did longer term experiment that maybe they left for, I don't remember, 70 days or so. And they didn't stop to leach. The leaching was linear, increasing. So I think that they don't stop. That's why I say that the values, the estimation I made for the plastic reaching the ocean, this is just for the plastic reaching the ocean, but all the plastic that is already there is constantly releasing carbon.
So, another question here.
Hola, buenos días. No es una cuestión, y perdona que lo haga en español. Lo que comentaste en un momento dado, que había unos 13 millones de toneladas anuales de plásticos que se iban al mar, de los cuales se degradan, y eso es lo que no estoy seguro de haber entendido, en el mejor de los casos 70.000 toneladas, es decir, menos de un 1%. Hay algo aquí que no me cuadra.
No, esto no lo entiendo, porque yo no he
dicho... No, es lo que me ha parecido entender, por eso te pregunto, es una pregunta.
O sea, yo no he dicho, la cantidad que se degrada no la sabemos. O sea, sabemos que entran 13 millones y yo lo que habíamos estimado era la cantidad de carbono orgánico disuelto que sueltan. Pero no sabemos qué cantidad se degrada. O sea,
de acuerdo, pues la cantidad de carbono orgánico que sueltan.
Sí.
Es decir, el resto, es decir, el 99 y pico % del carbono que hay en estos plásticos, ¿dónde va a parar?
Está en el plástico, o sea,
se queda en el plástico.
Sí, no se ha disuelto. O sea,
se van acumulando.
Sí, el plástico va soltando compuestos químicos de carbono disueltos y se van quedando disueltos en el agua, pero lo que no se suelta sigue formando parte del polímero y de los aditivos de la estructura del plástico. Entonces queda en forma particulada, digamos, podríamos decir, porque queda como en el plástico. Sí, no sé si...
Esto, es decir, se está acumulando, ¿no? Estamos hablando de que hay 13 millones de toneladas que se van acumulando con una pequeña pérdida del menos
de 1% anual. Sí.
Es decir, el crecimiento es de 13 millones de toneladas anuales, ¿no?
Sí, pero lo que se ha medido que hay en el océano flotando son al menos 250.000 toneladas. De esta acumulación, pero que tiene que haber mucho más, porque eso es lo que han estimado a través de muestreos que hacen en superficie, pero tiene que haber mucho más cantidad, porque si cada año entran 13 millones de toneladas, sí, hay mucho más. Lo que pasa que todo es que no se ha contabilizado.
Hay un agujero negro aquí.
Sí. Vale. De hecho, es que solo el 1% del plástico se ha encontrado. Ya. Eso es, sí. Lo demás se llama missing plastic, como plástico perdido, sí.
No está mal, vale, sí. Gracias.
Bueno, so If there is no more questions, perhaps we can leave it here. Thank you very much, Cristina, for this night.
Thank you.